Hi guys, welcome back to another episode of Family's Guide to Not Effing Up A-Level Biology. Today we're going to be doing glycolysis, the link reaction and the Krebs cycle. This video will effectively condense all of those three things that I just talked about. And after this, there's just one more topic, and that's pretty much all of respiration done and dusted. So if you've made it this far, then well done, you're doing pretty well actually. So yeah, let's get started with glycolysis. Well, actually, before we do get started with glycolysis, just like I did with the light dependent reaction video, I'm just going to go over a few key principles on this side. The first thing that I am going to point out is that respiration does not equal photosynthesis. Now, that seems pretty obvious, right? You're probably like, obviously we know that they're two different things. No, you don't, because I've seen so many people get confused between both of them, and it's not just because of the different naming systems. Like in photosynthesis you have NADPH, and in respiration you have NAD+, and it gets really, really, really confusing sometimes. So it's important to remember that we distinguish between both of these. Also, photosynthesis happens only in plants, whereas respiration happens in both plants and other organisms. And with photosynthesis, you're making storage molecules, where in respiration, we're actually using up storage molecules like glucose and, you know, all of your amylose and amylopectins that are hydrolyzed. Also, plants, most importantly, will carry out respiration as well. Sounds obvious because all of this stuff will happen in in the plant as well. So I think it's just important to get that into our brain before we get started with this video. Also, in respiration, we have two different electron carriers, and these are NAD+ and FAD+ as well. NAD+ is basically, the way I like to think of it is like, it's like the B-Tech version of NADP+. It essentially does the same job. It can hold 2 electrons, but this is the version that we use when we're referring to respiration specifically. So in its reduced form, it'll be NADH. With FAD+, so this is a different kind of electron carrier. Once again, holds 2 electrons. But with FAD, what it does is that it essentially carries lower energy electrons. With NAD, higher energy electrons are carried by that particular electron carrier. Whereas with FAD+, some electrons that have a lower energy level, those are the ones that are going to be taken up by FAD+. So in its reduced form, we will denote that as FADH2. Alright, I think those are the two main things that we need to know. Let's get into glycolysis. So, I think if we look at the word glycolysis in the first place, I'm just going to write it here. Glycolysis. Glyco refers to something like a sugar, and lysis in biology means the splitting of something, so split. So in essence, glycolysis refers to sugar splitting. So let's say the way that glycolysis starts is you have a molecule of glucose. So I'm going to denote that as a 6-carbon molecule over there. I'm just going to name that as glucose. And the next step that happens is that we phosphorylate that glucose. So we use 2 ATP molecules because ATP, it can be hydrolyzed into ADP and the inorganic phosphate. And the inorganic phosphate can react with a lot of different stuff. So in this case, we're reacting a few inorganic phosphates with the glucose. And what we're going to form is called glucose bisphosphate. And I'm going to denote the phosphate groups with this orange pen. P and P. So if we have 2 phosphate groups for 1 molecule of glucose, we're going to require 2 molecules of ATP for that. So I'm going to show it like this. So 2 ATP will go to 2 ADP. and the inorganic phosphate. And that inorganic phosphate, I'm going to do that in yellow just to go along with the flow. Inorganic phosphate, and that inorganic phosphate is going to start reacting with the glucose to form glucose bisphosphate. Now, glucose bisphosphate is unstable and it will start breaking down. So when glucose bisphosphate breaks down, we form two molecules of our friend triose phosphate. We've seen triose phosphate in photosynthesis. Don't get it confused with glycolysis. It's the same molecule, but I know we can make a few weird assumptions like I did when I was learning this topic, just trying to distinct both of them. So here we have our triose phosphates. Now, remember how many carbons triose phosphate has? It has 3 carbons. And that makes sense because glucose has 6 carbons. We're splitting glucose up. We have 3 carbons. Each molecule has 3 carbons. So in essence, we have 2 molecules of triose phosphate. So I'm going to write that down over here. There we go. And I'll just draw the arrows just to show that we have those triose phosphates forming. So in glycolysis, what we want to do is we want to make a molecule called pyruvate. And I don't think you particularly need to know too much about pyruvate, but we essentially want to form pyruvate from glucose. And in order to form pyruvate, we need to have a few more oxidation reduction reactions. So if I draw 2 arrows over here, and I'm going to draw out pyruvate here. Pyruvate also has 3 carbons, so I'm going to say 2 pyruvate. What will happen is that in order for it to form pyruvate, we actually need to oxidize the triose phosphate. Now, if we're oxidizing one species, we have to reduce something else. And what we're reducing is NAD+. Remember when we went over the light-dependent reaction video and we were talking a lot about why we were forming NADPH? This is going to be quite similar here. If you remember from the electron transport chain, we're going to see there's a very similar mechanism here in terms of respiration. So we need to form a lot more of those electrons that are being available in order to carry out a lot of those oxidation reduction processes. So for each triose phosphate, we need to reduce it once. We need to oxidize it once. So in that scenario, we need 1 NAD+ for each triose phosphate. So NAD+ will go to NADH. This doesn't involve photosynthesis, so no P's. And the same goes for the other triose phosphate. So NAD+ will go to NADH. In addition to that, the unique thing about this process is that we actually form some ATP out of it. I'm not going to go into the mechanism of why that happens, but in essence, you need to know that each triose phosphate, for its conversion to pyruvate, you actually form 2 molecules of ATP per triose phosphate. So I'm going to depict that over here. So 2 ADP plus inorganic phosphate will go to 2 ATP. And that's for one triose phosphate. We're forming 2 ATPs for each triose phosphate, so you have to do the same on the other side as well. So 2 ADP plus inorganic phosphate will go to 2 ATP. And then we finally end up with our 2 molecules of pyruvate. Now, one thing that you need to know for our spec is that we have a net gain of 2 different products. The first product we have is NADH. We haven't seen NADH being used anywhere else in glycolysis, and this is the first mention of it. It's not being reduced or anything. It's not being used up or anything. We're forming NADH. So we have a net gain of 2 NADH in total from the 2 triose phosphates. But we also have 4 ATP forming as a result of the 2 pyruvates forming. Now we have 4 ATPs forming, but we're using up 2 ATPs over here. Therefore, we have a net gain of 2 ATPs as well. So this is what the net gain effectively refers to when we're talking about glycolysis. Alright, that's the first part done. Let's talk about the link reaction. I'm telling you, it's a lot of mechanics, it's going to go quick. But yeah, so rub this out. The link reaction. Now, let's look into the name again. Link, it's clearly linking something up together. The link reaction. And in the link reaction, what's happening is that the the pyruvate that we've just formed is going to be actively transported into the mitochondrial matrix. Glycolysis, and I actually should have mentioned this earlier, so my apologies for that. I'm going to do it here. Glycolysis happens in the cytoplasm. Again, apologies for not mentioning that earlier. It happens in the cytoplasm specifically. So the pyruvate we've just formed, we need to convert it into something else called acetal, which is like, if you've done chemistry, it's like an acetal group, something like that. I don't know why I say it in a different way, but nevertheless. So we're forming, if I draw a cell over here, and this is like a huge mitochondria, we've formed pyruvate here. And that pyruvate will be actively transported, into the mitochondria. And this process is called the link reaction. So I'm going to draw out those molecules of pyruvate again. And I'm going to do one molecule of pyruvate just to simplify stuff. So pyruvate. Now, our final product is the acetal group. And acetal has 2 carbons. Pyruvate has 3. So clearly there's 1 carbon being lost, and that process is caused by something called a decarboxylation enzyme. So over here, one of these carbons is going to be removed, and that's going to cause CO2 to be released. Now, that chemical equation that we've been learning since we've started any form of science, glucose plus oxygen gives you carbon dioxide and water. That's where some of the carbon dioxide is coming from. So we've removed 1 carbon, which forms this acetal over here. In addition to that, we actually also form another molecule of NADH, because we need to reduce the we need to reduce the NAD in order to form NADH, and to reduce NAD+, we need to oxidize the pyruvate. Remember, oil rig, oxidation, loss of electrons, reduction, gain of electrons. So in this case, we want the pyruvate to lose its electrons, in other words, be oxidized, and as a result, the NAD+ will be reduced to NADH. So I'm going to write that in as well. And it's only for 1 molecule. So for 1 pyruvate, you'll have 1 NADH forming. So NAD+ goes to NADH. And it forms the acetal. Now, acetal just doesn't exist like this because it's very unstable and it can react with anything. So it will go around an enzyme, and that's called a coenzyme. If you don't remember enzymes, I think it's somewhere in topic 2, I think, somewhere there. So coenzyme here. And this coenzyme is called coenzyme A. You need to know that. So it effectively hugs the acetal just to make sure it isn't reacting with anything, because it's very unstable. And together, we call this whole thing acetyl-CoA. Now it's starting to look a bit familiar, isn't it? So one more time, the pyruvate is being oxidized to form to form acetyl. It's a decarboxylation enzyme is removing a molecule of carbon from the pyruvate, and NAD+ is being reduced to NADH. The acetyl group will combine with coenzyme A, and that will form acetyl-CoA. Cool beans. Oh, one more thing. Glycolysis happens in the cytoplasm. Please don't forget that. Alright. So the link reaction, remember it's actively transporting the pyruvate into the mitochondrial matrix. The last part of this video we're going to talk about is the Calvin? Wrong topic. We're talking about the Krebs cycle. Alright. Oh, it's a really long day. Right. Krebs cycle. The Krebs cycle happened in the mitochondrial matrix, so I'm going to write that down here. So, Krebs cycle. And happens in mitochondrial matrix. And there's an acronym for this. I think Dr. Shaggy and Mr. Greenberg came up with it. DINA for Anna. So I'm going to write that acronym over here. And as we're going through the cycle, we're going to talk through that acronym. So let me do a little box here. DINA. Let me just do it in a better way. Sorry about that. DINA, fa, na. Alright, we're going to start off with something, a molecule, and that's called oxaloacetate. Oxaloacetate, and as you can see, oxaloacetate has 4 carbons. What happens next is essentially the acetyl-CoA that we made from earlier. It's going to combine with the oxaloacetate. So, got too many pens. Alright, so CoA, that enters. Now the coenzyme itself is not going to be involved in this reaction. It's essentially just releasing that acetyl group. So I'm going to put acetyl here, and the CoA can buzz off and go back into the link reaction and essentially be recycled over and over again. So this is the first part. So oxaloacetate, a 4-carbon molecule, will react with a 2-carbon acetyl group, and we form a 6-carbon molecule called citrate or citric acid. Sometimes it's referred to as the citric acid cycle, the Krebs cycle, but you can just call it, well, that's pretty much why it's called citric acid in the first place, but let's just call it citrate, just to save a bit of time. And this has 6 carbons. Now, the next part of the Krebs cycle, essentially all of it, involves this whole acronym over here. The next molecule we form from citrate is called alpha-ketoglutarate acid. Now, you do not need to know that molecule's name, you just need to know that it forms a 5-carbon molecule. But I like to call it alpha-ketoglutarate acid because 1, it's fun to say, and 2, it puts a lot of context into what's going on. Rather than just putting a 5-carbon molecule and just moving on over there, it makes the cycle seem a lot more stable, for the lack of a better word, and it's just easier to memorize if I'm going to be honest with you, and you'll find that out in a bit. So, another arrow, and it's a 5-carbon molecule, alpha-ketoglutarate acid. Sometimes it can be shortened to ketoglutarate. Both hinterland for you. And that's a 5-carbon molecule. That involves the first part of our acronym, D and N. The D stands for decarboxylation. The N stands for the formation of NADH. So in this case, the decarboxylation makes sense, because we're going from 6 carbons to 5 carbons. So 1 molecule of CO2 is being removed. And in any case where CO2 is removed, it's a decarboxylation enzyme that's involved. And the actual process itself, pretty self-explanatory, is called decarboxylation. In addition to that, we are forming 1 molecule of NADH from NAD+. DNAR, the rest of the acronym is going to be all in one fat step. So it's going to involve decarboxylation, the formation of NADH, so DNAR. For ANA, that's the next part we're going to go over. So I'm going to make another arrow that goes back to oxaloacetate. The first part, D, so that seems to tell us that's another decarboxylation enzyme that's being involved. So CO2 is again exiting the cycle. So that's our D part. The next part is Na. So we're forming another molecule of NADH. NAD plus goes to NADH. So that's our Na. The next part is a Fa. Remember all those electron carriers we were talking about? There's a special one called FAD plus. FAD plus carries those lower level energy electrons. So in essence, we're forming FADH2, which is in its reduced form. Whenever we're using this acronym, and in the Calvin cycle in general, we're always reducing the oxidized form of the electron carrier. So in this case, NAD+, FAD+ is always going to turn into the reduced form, which is NADH or FADH2. So according to our acronym, we have reached the Fa. So the next step involves forming, or in other words, reducing FAD+ to FADH2. That's our Fa part done. The next part is R, and that is referring to the synthesis of ATP. This synthesis of ATP includes something called substrate level phosphorylation. Substrate level phosphorylation is when a phosphate group is donated from a donor molecule. That's essentially what you need to know. So that ATP over there is going to be formed from substrate level phosphorylation. So ADP plus inorganic phosphate forms ATP. And I'm just going to denote that with substrate level phosphorylation. And the last part is a NAR, so we're going to be forming more NADH. So, my green pen. Finally, NAD plus goes to NADH. And we've reached back to oxaloacetate. So, welcome to the Krebs cycle. It's definitely not as bad as some people make it turn out to be. Ketoglutaric acid, you see why I used it there? Because if I just used 5-carbon, it doesn't really work as well. And it looks like a complete cycle now. So we're going to go over it one more time. A 4-carbon molecule of oxaloacetate reacts with a 2-carbon acetal to form a 6-carbon molecule of citrate. Coenzyme A from the acetyl-CoA detaches from the acetyl group. The coenzyme A now returns to the link reaction in order to be recycled and used to bind with another molecule of acetal. The citrate now gets decarboxylated by a decarboxylation enzyme, and it also gets oxidized, which causes NAD plus to get reduced to NADH. This forms a 5-carbon molecule called ketoglutaric acid. Ketoglutaric acid is once again decarboxylated. It's decarboxylated. It's also, apologies, it also triggers the conversion of NAD plus to NADH, which is its reduced form. It also causes FAD plus to be reduced into FADH2, and another molecule of NAD plus is also reduced to NADH. So that's essentially the DNAR for RNAR part of what's going on. Remember that substrate level phosphorylation there? ADP and the inorganic phosphate react to form ATP via substrate level phosphorylation. Do mention that we're forming two NADs, NADH here. And then finally we return back to oxaloacetate. Alright, so the next video is going to be the last video on respiration. We're going to go through oxidative phosphorylation. Sounds a lot more scary than you actually think it is. And finally, glyconeogenesis, the weird stuff. Alright, I hope that video was helpful and I'll see you guys in the next one. Bye.